Wow, long time no see. So today we're actually going to talk about special senses instead of what we've been talking about. Remember, it's all part of this nervous system, and I want you to also remember that the specialized senses are basically they're found in the head. You might think about it; it's your eyes, your ears, your nose, and your tongue. So those are the basically the locations of those special senses. They are going to encompass a variety of different types of receptors, but they're specialized because they're only in that location. So what do they actually identify? They identify smell, taste, hearing, and vision, and equilibrium. So when we actually look at it, you have specialists that are involved in it. So when we're actually looking at the specialized senses, the first one we're going to talk about is chemical or chemos receptors. So where do we actually have them? We have them olfactory and we have them gustatory, which means that we're looking at smell and taste. So we're looking at the nose and the tongue. So one of the things that you want to think about is they go to the limbic system, and smell is one of the strongest emotional reactions that you get. Now, I want you to think about this. Memories are actually based upon previous experiences. So if you smell something, whatever the smell is, you develop a memory of that. This is one of the reasons that if you actually were to come in contact with people when they go to a funeral, a lot of times they smell saddlewood at the funeral parlor. And that's one of the things that you would actually indicate that if you smell saddlewood at other times, it may bring up those emotions that you had at that funeral. Or if you had, let's say, a certain particular flower during Valentine's Day, you might actually think about a reintroduction of those emotions based on that odor. So you have to keep that in mind. When we actually look at olfactory, as you guys remember, that was from cranial nerve number one, which is basically a sensory only, and it actually is when we're looking at it anatomically. Basically, it actually has a pathway that goes right back into the basal stem cells. Okay, for the receptors, and one of the things that we actually have is a first order type of pathway, and they are bipolar neurons. So if we were to look at it, here's a nice image of our nose. So if you're looking at this, for every square inch, it has about 10 to 100 million receptors. And it will cover there are three types of receptor cells. But basically, what happens is they go through the cribriform plate or in through the foramina of the ethmoid bone and in the cribriform plate, and basically they actually you can actually see them going through. And if we looked at it in more detail, it would look more like this. So when we actually look at the olfactory receptors, we also have supportive cells, which are epithelial cells. We have basal cells, which are stem cells to replace those olfactory cells monthly. And we also have olfactory glands which produce mucus so that we actually have moisture, so that the odorant, which is what we're smelling, can be converted into a liquefied version so that the odor can then get indicated by those receptors. Um, Physiologically, what happens is we have the ability to actually have hundreds of scents. So when we're actually looking at it, and this is one of those things that we notice, we can have a slow threshold for sense. So you can smell things when they're very small. This is one of the reasons that you might notice odors that are you come in contact with. Um, here's one of those indications that I always remember. I remember, you think about this when you actually smell, let's say, gas. you know, you have gas in your home, maybe from the stove or something like that, and you pick up small odors of that. So when we're actually noticing it, this actually gives you a good indication of what's going on that may have changed. And that's good as far as evolutionary wise, because if you're thinking about it, if you were going to eat something as as one of the early humans, odor was a good indicator to help us understand what was fresh or what was not fresh, or what was still viable as far as food or not, if it had spoiled. So this is one of those good things that we actually see. And it only takes a small amount of molecules to actually indicate that odor. So that's very useful for us. There are also indications that when we actually look at it, it goes into our sense and it actually emits an emotion. So one of the things that we also see is that when we actually look at it, there are people that do not smell odors quite as well. They're known as hyposemias. And a lot of times what happens is over the age of, after the age of 65, you actually start decreasing your ability to actually smell those odors. Not only that, but if you actually have neurological changes or you actually consume drugs, or if you're a smoker, when people smoke, they actually also get that smoke into their lungs and areas like that, and it actually decreases their ability to distinguish odors. Now, here's one of the things that you may not think about. Most taste Most tastes are also integrated with the odors. So what happens is your tastes are only four different types of basic taste: saltiness, sourness, bitterness, and sweetness. Everything else gets migrated up into from our mouth up into our nose, and in the nose it actually picks up those other odorants. And what happens is you take those two types of chemical detections, and it goes into your brain, and that's where it actually emits those memories that you've had, that interprets what you actually consuming. So when we're actually looking at it, people that actually smoke, they also have a decreased amount of an ability to actually distinguish differences between different types of tastes based on the odorants. So when we're actually looking at it, the odorant actually changes the sodium channels and it depolarizes, and we actually emit a nerve impulse. So when we actually look at it, we can adapt and we can actually have a threshold. So the odor threshold is very, very low, but our adaptation is rapid. So what happens is we actually adapt and we actually get rid of that odor in a very short period of time. So if you actually smell something, you can actually have that odor go away and you become more familiarized with it. Think about this, and I want you to consider this. You're going to a restaurant or you're in the environment of a restaurant. And in that environment, you smell the odors of the cooking, and it enhances your experience. And you actually come into that restaurant and say, "Ooh, it smells so good," and all that. Give it a couple minutes, let's say 15 minutes, and you don't notice the odor anymore. This is what we're talking about. The odor adapts rapidly, and you don't notice it as much. So when we're actually seeing it, this is one of those things that we see. The low threshold actually allows us to actually pick up on that odor quickly in a very small quantity. This is one of the reasons that when we actually smell natural gas, they actually have added in actually methyl mercaptan, which is basically an oil, which is actually helping us to actually distinguish the presence of gas. So when we're actually looking at the pathway is basically goes in through our foramina and then goes in the olfactory tract and then to the olfactory primary olfactory area in the temporal lobe. And then it can actually lead us to other things where we actually identify that odor from the past. Gustatory, on the other hand, is the taste. So is our taste of our sense of taste. Basically, what happens is, like I said, we have our primary tastes, which are sweet, sour, bitter, and salty. Now, there has been some speculation that there's a fifth one, and there's different names for it. Sometimes it's referred to as savory, and sometimes it's referred to as um, stale. So when we're looking at it, there hasn't been a real consensus on that. So I'm not going to expect you to know that. So the four tastes you should know. So that's one of the things that I want you to recognize. So when we're looking at it, and this is one of the things that they have also speculated, that sometimes we actually have the gustatory things on the different areas of the tongue, Well, don't worry about that because there's no pattern that is actually established that is concise and clear cut for every individual. So it's better not to actually get a misinterpretation of where the tastes are, which taste is where. So when I want you to realize is that basically we consume things and we actually identify these tastes. But there's only about 10,000 taste buds. And that means that you have less ability to have taste. Now keep in mind, things towards the front of the tongue will be more like the bitter. And I want you to notice that that's more. Why? Because basically, bitter is something that is usually associated with poisons, medications, things like that. So you want to reduce that. Whereas one of the things that we actually see is sweetness and saltiness are usually things that we actually pick up more of. And so that's why you'll notice it adapts. And a lot of times people will actually get to a certain level of saltiness in their diet or sweetness in their diet and it actually adds and adds and adds and they still don't have that threshold. And so what happens is a lot of times people will actually, and you'll notice this in most of the food that we actually get that are prepared foods like frozen foods or canned foods or even in a restaurant, they add quite a bit more salt and a lot more sugar to those foods items. So when we actually do look at taste, for instance, I take my cup of coffee, and what do I taste in there? I can taste the bitterness of the coffee. I can taste the sweetness of what I added to it. I can taste the sourness, and hopefully, I don't taste too much saltiness. But you never know. You have to watch. So, mm, tasty. Anyway, so when we're actually looking at the taste buds, they basically look like onions. And they actually have hairs, which are gustatory hairs, and they basically actually will you'll notice that you can actually replace them every 10 days. So if you ever burn your tongue, and you probably have burned your tongue maybe on hot cocoa or something like that, what happens is you reduce your ability to taste, but then what happens is it gets repaired and gets replaced within a short time. When we look at the physiological aspect, like I said, the threshold we have of our taste, basically the most sensitive will be things like bitter, because most of the time bitter is related to poisons, which are also, if you look at medications, typically they are poisons. So you actually have a more sensitivity to it. Saltiness and sweetness, you're less sensitive to it. So what happens is to adapt, you actually may want to have more of the salty or sweetness. And you'll notice if you eat salty foods, you may notice that over time you may actually not notice the food at all unless it has a certain level of saltiness. So, but it is a first order um, neuron. When we actually look at it, we actually can see that we have different cranial nerves that are involved. I'm not going to ask you that at this point. Hey, calm down, buddy. Um, but it does actually go to the limbic system, and it also is involved with your conscious perception of taste, which gets us to our next group: vision. Vision is in your eye. I hope you can see it. But if you don't, that's okay. We're going to go over the information on it. And I want you to realize that one of the things that we see is that basically we have receptors. Now, one of the things that we see is this. In the eye, if you're looking at the eye, it actually is protected by eyelids, and we have glands, and we have a conjunctiva, which is basically a membranous structure that basically goes up to the edge of our cornea. Now, when we're looking at these accessory structures, they are to help the eye to protect it. And if we actually look at our eyelids, they basically, most of the time, most people will notice that they close their eyes during sleep. So it protects it from any kind of damage or things that can fall into it. So if we also look at your glands, they're also there to lubricate the eye, and the conjunctiva is membranous. It's basically a mucous membrane that is actually there to actually help also. You also have eyebrows and eyelashes. These are actually there not just because they look nice, even though most people think about them as a way of keeping, having them make our eyes appear a certain way, whatever that may mean for the individual. But if you're looking at it, the real purpose of it is to prevent things from falling into the eye, like objects, perspiration, things like that. And if you, wow, look at that person's eye. Wow, see it? Um, did you ever find somebody was looking at you? There you go. We only see about, it's only about 1 inch diameter, but basically you only see 1/5 of it or 1/6 of it. The rest of it is protected by the orbit. So I want you to realize that. So structurally, that's one of the things we see. Now, we also have the lacrimal glands, and they basically are above the eye. As you notice in this diagram, we produce about 1 milliliter of tears a day, which actually has lysozyme in it. As you might remember, this is a very highly effective um, um, enzyme, which is hydrolytic, which means that it can destroy cells very readily and very well. The only time that it doesn't is when there's too much of a particular bacteria, and most of the time it's bacteria that actually gets into the eye that has overwhelmed the effects of that enzyme. So what happens is we can get conjunctivitis, and we also have we also have some extra ocular muscles. We have 6 pairs of them. You'll notice that they're known as rectus muscles, which will be superior rectus, inferior rectus, lateral rectus, medial rectus. And I think we've talked about this in class at some point in time. So basically, if you're looking at your eyes, you have your lateral rectus, which will move the eye this way, but then the other eye will use the medial rectus to move the same direction. Most of us move our eyes in direction. If we have the superior rectus, we move the eyes upward. We have the inferior rectus, it moves it downward. So this is what we're actually looking at. We also have 2 oblique muscles, which basically rotate the eye. So there's an inferior and superior um, obliques. Um, so when we're actually looking at the eye also, one of the things that you might notice is that we have basically 3 layers of the eye. We have a fibrous layer, which is known as the fibrous tunic, which is the outer layer, the vascular tunic, which is the middle layer, and we have the nervous tunic, which is the inner layer. And we're going to break it down so that you actually have a better understanding of this. Now, one of the things that I'm going to point out is that we will actually have a couple diagrams on the test. One of the diagrams will be an eye diagram with the different parts. So I do think it's important to go through the different aspects of the eye so you understand how to do the diagram. So if we looked at the fibrous tunic, which is basically that first layer, we will actually notice we have the cornea and we also have the what we're going to also look at in a minute. So this is the portion that is more fibrous to actually hold the structure of the eye. Now the cornea itself only is that clear portion that's at the front or the most anterior portion of the eye, and basically it's made up of we're not going to worry about the actual specifics of it, but it's basically epithelial tissue and some connective tissue. But one of the things that is important about it is that the cornea is really the thing that actually affects our vision as far as astigmatism, because maybe one side is thicker than the other side. So I want you to realize that. We can actually transplant corneas because there's no blood vessels. And because there's no blood vessels, there's no antigens to reject it. Um, Also, part of it on the latter portions and the more posterior and lateral portions is known as the sclera. That's the white of the eye, and this is made up of more fibrous portions, fibrous connective tissues, which actually gives it more structure. Um, the next layer, the vascular layer, or the vascular tunic. What you'll notice is we have the choroid and we have the ciliary body. Now, they do connect together, but basically, this is the portion that actually has blood vessels in it. This is why you actually notice the blood vessels through your sclera. And the more anterior portion, which is why if you're actually looking at this part of the eye right here, where I'm trying to mark off in this coloration, this is where we have the ciliary body, which is made up of, we have muscles, ciliary muscles, and that actually affects the lens of the eye. So I want you to realize that that's one of those things that's rather important to understand. So if we go on, what we also notice that our next layer is our where we actually have the vascular tunic actually affecting our iris and our pupil. Now, one of the things that you'll notice, the iris, if we actually were to look at right over here, this part and this part, which I'm coloring in right now. Oops. Let me try to change the color there so that you guys can see this a little bit better. If we actually were to look at this part right here, this is our lens. But the greener portions are actually where we see the ciliary body connecting up to the iris. And basically, the iris is basically the coloration portion of our eye. That's the part that we actually see and say, what color eyes do you have? And the lens is behind it. And the lens is what can be actually used to focus on an object, or what we refer to as accommodate. So I do want you to think that that's one of those things that's useful for your understanding. So when we actually look at the iris and the pupil, when we're looking at somebody's eye, we basically see the iris, and the pupil is that space that allows the light through. And it can actually change. What happens is the change in the dilation or the constriction of it is based upon light. It can also be related to medications. So I do want you to realize that. So when we look at the lens, though, the lens is basically just an onion-like structure. It, if you were to dissect the eye, you'll notice that it looks almost like a piece of plastic, even though it's not. And basically, it can be used to actually bending it and stretching it to actually accommodate, which means that we're trying to actually get focus on something. So, if we were to look at it in more detail, what do we see? Over here, we actually see the ciliary muscle and the ciliary process, which leads to over here, which is our iris. Okay, see that? And we have the lens. Now, in front of the lens, we have the anterior chamber, and behind the lens, we have the posterior chamber of the aqueous humor, which is something you should know. Okay. Why am I mentioning it? Because we also have outlets over here, which I'm trying to find right here, the sinus, which is basically where we actually get some of that fluid leaving the aqueous humor. Now, why is that? So let's go on and look at what we're actually also examining. So if we're actually looking at our third layer, that's our nervous tunic, which is where we see the retina. Three-quarters of your eye have the retina, which means at where we actually see the iris, the pupil, and the cornea, there is no retina. What you'll also notice is we have a couple structures that are important. Now, this is a normal view from using an ophthalmoscope. So when you're actually looking at it, you'll notice a couple structures. You'll notice what we actually have as is known as the optic disc. This is the area at the back of the eye. Now, notice the optic disc is right here. And if we looked at it over here, it's right in this area of the eye. So what happens is that's also known as the blind spot. So I want you to realize that that's important to know. What you'll also notice is that we also have, but that's not directly at the very back of the eye. If we go straight back to see the back of the eye, we would notice it's offset, and that's where our optic nerve comes in. That's also where the blood vessels come in. That's one of the reasons that the optic nerve disc can also, or the is also known as the blind spot. And you'll see the blood vessels coming out of there. Now, we also have what's known as the central fovea, where we actually have the macula lutea, or what's also referred to as the macula densa. Basically, what happens is that area is at the central portion at the very back of the eye, like I drew over here, and that's why it's called central fovea. But why is it referred to as macula lutea is because, or macula densa, is because that's the highest concentration of receptors. Now, there are problems that can exist with the eye where we actually have problems like diabetic retinopathy, which is basically where the retina pulls away from the back of the eye and it starts floating inside. And you may actually have sometimes there's blood vessels that actually have some sort of a um, hematoma or a blood clot that starts floating around. So that's what's referred to as floaters. But what you also may notice with diabetics and people that have hypertension and other problems, including trauma, like it says over here, boxers, what happens is it can actually cause the retina of the eye to detach. And if it detaches, the person will have an inability to visualize. So, most of the time, that actually will cause a condition that's known as where we actually have blindness. So, Um, now, so when we actually look at the receptors of the eye, we have two types. We have rods and we have cones. Rods actually identify black and white. So, and cones detect color. Now, what colors do we see? We see red, blue, and green. So there are three types of cones. So I'm going to put that there so you guys can see that. All right. So when we're looking at it, they are the red, the blue, and the green. Now, if you think about it, some of you may remember years ago before the invention of these flat screen TVs, you had TVs that were color TVs that you could actually adjust the colors. Now, if you're looking at those adjustments, they're very similar to what we actually have on the eye. You have the rods that detected black and white, so that was your contrast, and the cones detected the three colors: the red, the blue, and the green. And it's the combination that would allow you to see various colors. Now, one of the things that you would also notice that basically you have more cones overall than, more rods rather than cones. This is one of the reasons that you can actually see more, you can see light in dark or dim areas. But you need to have the cones. The cones are basically more around the macula lutea and the back of the eye. So what happens is you can see light that has color, but you can only see it during lighter or brighter time frames where you see light rather than darkened areas. This is one of the reasons that you might have heard that cats actually see things in darkness. Well, the reason that we know that is because we look at their receptors, and we've actually identified that they have more rods than they have cones, and it's more distributed throughout. So that's one of the reasons that they actually detect more of the light and dark, even in dim areas. So the photoreceptors are basically the sharpest at the fovea centralis. When we actually look at the retina, though, here's one of the things that I want you to see, which may actually help your understanding. Notice, and I want you to notice this right here. Over here, what we see is this. This structure right here is actually the portion of the 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 back of the eye, the retina, where the light comes into it. So basically, your light goes in from that direction. And what happens is, as the light passes through, it goes past these different layers, which means you actually have different layers, and then it hits the receptors right here and right here. So when we're actually looking at it, we actually have the receptors that are in what's known as the where we actually have the neurons, which is where it's detected. But then the signal goes into these other locations where we actually have the bipolar cells, and then it goes into the horizontal and the endocrine cells, where we actually have it then transported back to the optic nerve. So when we're looking at it, this is one of those things that's important to understand. So when we're actually looking at it, the rods and the cones, basically we actually have 120 million rods, and they're distributed throughout the eye, whereas we only have 6 million cones. And what happens is that's why color is not as clear or as sharp as you might find black and white. What also happens is, and we talked about the pathway, basically it goes into the bipolar cells to the ganglion cells and then gets into the basically the optic nerve to go then to the primary visual cortex at the occipital lobe of the brain. So when we're actually looking at it, one of the things that also happens is when we get that light, the light has to be clear. So basically, we use our lens to accommodate, which basically means that it actually adjusts to actually allow us to visualize the light in a focused manner, where it has to hit the retina correctly. But it also relates to what happens as far as in those clear areas. Now, one of the things that happens that may also occur over time is that maybe some people will actually lose their elasticity of the lens. And so what happens is we lessen our ability to see. That's known as presbyopia. Presbyopia happens to a lot of people that are elderly or older, and they just don't have the ability to see. The other thing that happens, which is not also clearly known also by a lot of people, is that over time, your perception of color changes. So when we're actually looking at color, as you age, your lens get because of your aging process, it gets more yellow. And as it gets more yellow, what happens is your perception of color changes. So, one of the things that we also have to look at those clear areas, those cavities, those spaces of fluid that are inside the eye. So, the one that's in front of or the frontal portion is known as the aqueous humor. And in front of the lens, we have the anterior chamber. And in the chamber behind it is known as the posterior chamber of the aqueous humor. But behind the lens completely and behind the 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 majority of the lens, the eye, we actually have the vitreous humor. Now, the vitreous humor has more of a jelly-like consistency. It doesn't have a more liquidy or watery consistency. The aqueous humor has more of a watery consistency, and it's replaced every 90 minutes. The aqueous humor. But the vitreous humor is what's formed when you were an embryo. So this is one of those reasons that when we actually get a floater or we get other types of things in it, it sometimes floats around and doesn't get and it blocks our vision. If it actually, it's sort of like if I put my hand in front of the screen, what you're seeing, you see I'm there, but I'm blocked. And this is basically because there's something that's been moving around, and some of you may experience floaters, and that's basically what a floater is in the eye. Now, in today's level of surgery, optic surgery, a lot of times I've heard that they are able to do some things about that. Um, so that's something important to understand. Now, one of the things that happens is to maintain the structure of the eye, you actually do have intraocular pressure. And that intraocular pressure allows for the eye to have maintained its structure. But unfortunately, there are some people that increase the pressure. Now, one of the reasons that that happens is it may be related to the aqueous humor is not draining properly. And it can also occur over time, and it can actually also cause that pressure increase. So the eye gets more pressurized, and because of the more pressure, it's not able to actually expand that much beyond a certain point. And so what happens is once the pressure is increased on the inside the eye, excuse me, it can actually cause the retina to detach or to create more floaters. And when it does that, it can then cause blindness. So when we're actually going back to the aqueous humor, remember this image? Over here we have the the sinus. Well, that's where we actually drain the aqueous humor, and the fluid goes out. Okay. So when we're actually looking at it, what happens? A lot of the image relates to three basic processes. What is the refraction of light? So I want to try to give you some some background as far as the light physics. So what happens is when we have light and it goes through an area, it bends. So that's what we're looking at when we're looking at the refraction of light. So when you're passing light through your cornea or your lens, it actually bends the light. When we do accommodation of the lens, which means we change the shape of the lens to actually focus the light. And when we actually are looking at the pupil is the third thing. So the pupil will actually change its amount of light that actually goes in or doesn't go into the eye to hit the retina. And that will also create an ability or an inability to visualize the images that we're looking at. So this is a visual interpretation of the refraction light. Notice the light goes in, it bends, and it changes the actual direction of the light. Sort of like what happens when you actually look through a fishbowl. The refraction of light will actually bend the light, and the thing is that we want to actually have the light, and most of the time when we have this refraction, just like what we saw in the microscope, it flips the image upside down. So when we're seeing it in the lens, it does the same thing. And 75% of the refraction occurs in your cornea. So the rest of it is based on what goes on at the lens. The accommodation of the lens, on the other hand, is how do we bend the lens? Now, you'll notice that one of the things that we have to do is we have to stretch it out, which will make it narrower but elongated, or we actually reduce the stretching out from the muscles, and it allows the muscle or the lens to bulge up. So this is what happens. And basically, a lot of times what we do is we can also have accommodation of this by using external lenses. So, what do we actually see? Well, one of the things that we see is for most of us, when we actually look at objects that are close by, we use 4 inches when we're a young adult. By the time we're 40, we're now looking at 8 inches. And a lot of it's because of that reduced elasticity of the lens. And when we get older, it actually gets a little bit more difficult. So that what you're seeing is people over the age of 40, sometimes they need eyeglasses. And that's to help them see things close up, which are different than when they were younger. And keep in mind we also have presbyopia also happening. Okay. So when we're looking at nearsightedness, that's where we actually see things close up, but we're not able to see things far away as much. So that's myopia. Farsighted is hyperopia, which we can see things far away better than we can see things close. And one of the other problems with refraction is astigmatism. As we talked about earlier in this presentation, astigmatism is where we actually have one side of the cornea is thinner or thicker than the other side. So that creates an inability to have the symmetrical aspects of that lens. So when we're looking at the correction, though, notice here on the top we have a normal eye. Over here on the left, we have nearsighted, so myopia, where we actually are focusing it, and but hits before the retina, whereas hyperopia, it falls um, behind the eye. Okay, all right. So what happens is when we're looking at these two images, we're looking at the comparisons. On the right, we're actually now adding in the lenses. So on the nearsighted, we actually have to have a concave lens to actually refocus it so it actually hits the retina, as opposed to a convex lens, which is used to help with farsightedness. So we can actually notice that. Okay, so knowing myopia as nearsighted, hyperopia as farsighted, and astigmatism can be useful and will be asked on the test. So, one of the other things is we actually have changes to the pupil. So, the pupil can actually get constricted, which we reduce less light through. And a lot of times it sharpens your image, especially when there's brighter light. Now, notice when we also have it, I want you to notice that when you're thinking about it, you guys might remember I keep talking about when you're actually, let's say, in a bright light area. You go to the beach, you go skiing, and there's the slopes. Well, you have to constrict your pupil so that you actually reduce the light. Otherwise, you're actually having an overwhelming amount of light going in, so you're not able to see details. So that's important to realize. But you also may notice that when it gets darker, you dilate your pupil, and so it allows more light in. But medications can also be used to actually change how your pupil reacts. That's why when you go to the eye doctor, they usually put eye drops so they can see without having any problems into the eye. The other thing that happens with your eye is you have convergence, which means because you have your two eyes, it allows you to see objects with an overlap. Now, keep in mind, we actually have a blind spot in each eye, which is where we actually have the optic nerve. So, what happens is perceptually, our mind actually fills in those spaces, but part of it is also because of the convergence that allows us to fill in those spaces. Um, we also will notice that our eyes will move sequentially in most cases. Otherwise, you get a lazy eye. So, a lazy eye, you have one eye directed here, and the other eye is going someplace else. So, that happens. Okay. Now, when we look at the physiology of the vision, we have the rods and the cones. Now, here's an example picture of a rod and a cone. Notice they look like rods and cones. Basically, what we see is this: we have opsins, which are basically protein, and we actually get a derivative of retinal, which is derivative of vitamin A, which is, you might remember, when you were growing up, they probably said eating carrots helped you see better. Well, basically, it's because of the conversion to the retinal A. Okay. Does it mean it affects you as far as your vision, as far as ability to see? No. It just increases the ability to actually have the possibility of these particular pigments. But your lens will actually get affected in different ways. And I used to eat carrots when I was young, as a kid as a snack. And my eyesight, I still needed glasses. So it has nothing to do with the lens. So I want you to realize that. So when we're actually looking at the physiology, though, it basically creates the proteins, and then we get conversion. Now, what I do want you to realize is that with the rods, we get rhodopsin as the single type of protein, that the pigment, whereas the cones have three different ones, one for each color. Um, Now, when we actually look at them, they get activated, they can get bleached, and they can get regenerated. So, this is why we can actually see light and dark adaptations, where we actually notice the sensitivity to light as light changes. And when we look at it visually as far as a diagram, this is what's going on. We get the light stimulates it, it actually triggers it to actually get a response. That's basically what we're seeing. Bleaching will occur if there's too much, and we can regenerate it though. Now, one of the other things that happens is we can also get problems with this. So, one of the problems with the receptors is color blindness. Now, what is color blindness? It's the inability to distinguish different colors. Now, here's the thing, most of the time it's a red green uh, problem. In other words, people that have the colors of red and green, they can't distinguish red from green. Most of the time, color blindness is a genetic disorder and it usually affects males more so than females. So, one of the things that I want you to think about is this. Think about Vincent van Gogh, the famous painter. Look at his paintings, how colorful they are. Okay, so you thought about that. Great. Now, think about this. He was colorblind. And he was actually painting his paintings because he thought that's what the world looked like. He couldn't distinguish red from green. And so what happened was he was painting the world the way he saw it. Interesting, right? So I think that gives you some indication. And I want you to realize, even though it affects mostly males, there are some females that can be affected if their mother was a carrier and their father was colorblind. So I want you to understand that that does happen. Okay. Night blindness, on the other hand, is narcolepsia. Is basically you have difficulty seeing light, and this is referred to as a deficiency in vitamin A. So, and we can actually test for color blindness, and there's two test plates that are used. So I want you to realize that. When we look at the regeneration, we actually can see that it takes 5 minutes to regenerate, and it can actually take 90 seconds to regenerate a cone. So I want you to realize that. Um, but don't worry about it too much, those details. We can adapt to the light in darkness and light. As you might notice, it does happen. When we look at the darkness and lightness, it relates to the sodium channels. And when we actually see it, if we actually were to look at a neurotransmitter of the eye, you'll notice glutamate is what's released. So when we look at the visual pathway, it basically triggers the receptors. It goes to the horizontal cells, the bipolar cells, and then to the ganglia cells, which then go to the optic nerve, which then leads to the occipital lobe, which is where we have the primary visual cortex. Now, here's an actual photograph of a brain. Notice, this is what I want you to notice, just to get you a better perspective on this, that when we actually look at it, here we actually have where we have and I might as well change the color so that you're seeing this. Right over here and right over here, we have the optic nerve, which goes out to the eye. Now, when we actually are looking at this, you may also notice that the optic nerve will go to the optic chiasma, which is right here, and then the optic chiasma and goes to the optic tracts, which go back right over here, back to the occipital lobe. So that's something that may be useful for your understanding of why it does what it does. So I think that might be helpful as far as visually identifying the pathway. Okay. And we basically interpret the pathway or what we actually saw. You look at an object, you see what it looks like, and then you actually interpret it. And it's based on what you previously saw. So when we actually look at it as far as the fields, what you'll notice on this diagram, it actually shows you more of what you're actually visualizing from the colors. So look at those colors, look at how it looks, and you'll notice it does cross over. It crosses from the left to the right and the right to the left. So that may actually help your understanding. So most of the time, if you're really thinking about it, we're looking at the world and at the back of our eye, it's upside down. But because we're used to it, we interpret it in a certain orientation and visually identify it as being upright rather than upside down. Now, the last portion we're going to talk about is the ear. Now, if we look at the ear, this is what an ear looks like. So if you're looking at this ear, we're going to actually see it has three parts: an outer ear, a middle ear, and an inner ear. So when we're looking at the external ear, now look at this little diagram down here and compare it to our overall diagram. You'll notice that basically we actually have cutoffs where we see the different parts. So if you're looking at right here where we see this line, anything to the left of it is our external ear, which basically falls right along here. Okay? And then our middle ear also cuts off in a very similar pattern, but it cuts off basically where we see that blue area as opposed to this tan area, which is our inner ear. So let me try to draw a line so you see that cut off. Okay? And so let's take a look at it over here. So that's our cutoff. So at external ear, middle ear, inner ear. So we're going to talk about the different structures so that you have a better understanding of those structures, of where they actually are, so that you have better clarification of it. So when we look at that, and but let's talk about what the ear is used for first. Well, basically two things. You hear things and you use it for equilibrium. So our outer ear or external ear will actually collect the sound waves. How does it do that? From the pinna of our ear, this auricle, basically. And it collects it and goes in. At the eardrum, which is the beginning of your tympanic membrane or your ear membrane, is where we actually get the vibrations. And the vibrations come in and it vibrates the tympanic membrane or the eardrum so that we actually move three little ossicle bones known as the malleus, incus, and stapes. They're also known as the um, anvil, the hammer, and the stirrups, where they actually cause fluids in the cochlea to get vibrated, which is at the oval window, which allows the round window to be projected. And then what happens is the inner ear, which is where we see the basically it says labyrinth, but basically at the labyrinth we have the cochlea, where we actually have the receptor cells that can get stimulated. So when we're actually looking at the external ear or the outer ear, basically we have the auricle, which is basically the pinna. It's made up, and you guys know about it. A lot of people pierce it, and it basically narrows like a microphone. It collects the information into this narrow canal, the external auditory canal, so that we can actually get the sounds in. The canal actually has, if you recall, it has ceruminous glands which produce cerumen, which is earwax, and it basically funnels it in there so it can actually stimulate the tympanic membrane. So Basically, hopefully everything's in place. Um, but basically, what happens is it then creates the movement of the tympanic membrane or eardrum. Now, one of the things you'll notice is that the you can actually get perforation of the eardrum. Now, what will happen is you can actually get hearing loss, pain, ringing, dizziness, all kinds of problems with it. Now, how does it happen? Basically, you get an ear infection. You you get an explosion, and it causes too much of a vibration. You get scuba diving, and that can actually perforate it. Now, here's the thing: when you actually perforate it, you lose your ability to hear. So I want you to realize that. So to help you understand that, I'm going to actually and notice that when we actually get infections, this can also happen. So a lot of times when we get infections, you actually have loss of hearing. Also, a lot of times this happens. So we'll talk a little bit more about that, and I'll try to give you some images of an ear so that you have an understanding of what it looks like. Um, so Now, here's the middle ear. Now, notice the middle ear, we actually start with the tympanic membrane. We have the three bones. We have the malleus, the incus, which is right here, and the stapes, which is right there. And this actually is on the oval window, which projects onto the oval window so the fluids inside the cochlea can move around. And then we have the round window over here, which allows a pressure valve so it doesn't actually get pressurized and not releasing the amount of pressure. So that's rather important. So it's basically causes the vibrations to move the bones, and the bones can actually then move that round window, oval window to create the round window movement. Now, one of the things that you might also notice though is that you can also have arthritis. So a lot of people don't even think about it, but you can have arthritis between these bones. But it's in the middle ear is where you usually notice infections. It's not the external ear. So I want you to realize that. So when we actually look at the labyrinth, which is where we actually have the structures, this is where we see the cochlea, and we have the semicircular canals. The cochlea is specifically for identifying hearing. The semicircular canals is related specifically with equilibrium, which we're going to talk about in a little bit. So when we look at the labyrinth, it looks like this. Notice the cochlea and the semicircular canals. Now, the semicircular canals are in three different directions. So I want you to realize to actually understand the directions of balance or equilibrium. So when we're actually looking at it, they are connected at the labyrinth, okay, where we actually have connections of different things, more detail wise. But I don't want you to worry about that part. So, and this is just going over some more details of it, but I want you to realize that when we're actually looking at it, the real key things that you want to really look at, the bony labyrinth is the bone in the temporal bone, whereas the membranous labyrinth is the actual structures within it. So I want you to understand that that's where we're seeing. Okay. Now, what do we see? The semicircular canals are basically going to have the three directions, like I said, and the three directions will be useful to our understanding the balance. So let's look at this picture one more time. Now, notice here we actually have the semicircular canals. Watch it, buddy. I know you got woken up. I know you did. Um, yes, I know. Um, yeah, my dog. So what we see is we have nerves that are on the area where we actually have the semicircular canals, which is the basically the vestibule branch of the vestibular cochlear nerve, and then we have the portions that are connected to the cochlea, which goes into the cochleal branch. So that identifies hearing now. So when we're looking at it, we talked about the cranial nerves. When we actually look at the cochlea, though, this visual really does give a good representation of what's happening overall. You get the vibrations from the outside, and here's one of the things I want you to think about. The Remember we also heard about Vincent van Gogh earlier. You guys might remember Vincent van Gogh. At some point, they claim he got mad and he had an argument with his girlfriend and he cut off his ear. Well, basically, what he did was he didn't cut out all the details inside. All he did was cut off the pinna of the ear. And I want you to think about this. As you know, artists are sometimes very emotional. And really, what he did was he only cut off this outer portion, which reduced his ability to funnel the sounds into the external auditory canal. And so what we're looking at here is the external auditory canal actually has the projection of the sounds, which then vibrate the tympanic membrane, move the bones, and cause pressure on the oval window to cause the fluids to migrate all in this section to actually come and get a pressure valve at the round window. Now, as it does that, it also causes, and you notice this cochlea is actually expanded out as one tube appearance. But in between, which is going here to here, in between, we actually have structures where we actually have cilia and we have what's known as the basilar membrane. And it's the membrane and the tectonic membranes which will actually move and cause the cilia to actually move and get stimulating of the nerve. So when we're actually looking at it, these vibrations will actually cause an effect to the cilia or microvilli. And so when we're looking at it again, just notice that this is what we're seeing. Okay. So in a cross section, it basically has the tympanic structures occurring. So I'm going to skip over this slide so that you can see it. But this is the one that you really want to look at. Now, if you remember back, we actually had these tubes. Now, this and this are the two portions of the tube. Here, which is what I want you to point out, right in here is actually the portion where we actually have those two membranes, the basilar membrane and the tectonic membrane, which is where we actually have the receptors. So if we were to look at it in more detail here, you would actually notice that we actually have those microvilli or cilia, which are there to actually allow us to actually get the sounds. Now, when we actually look at it overall, we get the compression, the vibrations, and the vibrations create a certain frequency, and those frequencies allow us to then identify those frequencies. So, anatomically, the basilar membrane is basically related to what's called the organ of Corti. And it's the organ of Corti, which is where we actually have the hearing organ. Okay, that's really what it is. And the tectorial membrane is more delicate, and that is also related to the hair cells or the cilia hair cells. And it's in a gelatinous membrane. And basically, what happens is as they move, the locations of where it is in the cochlea will determine different pitches. And what happens is that means we actually get a different identification of the, the sound. So, the frequency of the sound in particular. So, when we're looking at it in more detail, this is our organ of Corti. Notice this is our tectorial membrane. And over here we have our basilar membrane. So let me just try to give you more visual. So this is our tectorial membrane. This is our basilar membrane. And right about here, you might notice the little cilia, the hair bundles, basically is what's referred to as. And so that's where we actually get the stimulation that then triggers the neuron. the hair cell to get stimulated. So that's basically what we're actually looking at as far as where things are going to occur. Interesting. And there are 16,000 different hair cells with 30 to 100 microvilli associated with it. Now, those numbers are not really that significant for you to remember, but it gives you a perspective. And when we look at it, sound, typically what we see is we can actually hear things. We have a range of frequencies of 1,000 to 4,000 hertz, which is how many cycles per minute. And we can actually have some individuals that have a wider range, which means 20 to 20,000. But speech is between 100 and 3,000 hertz. The amount of the frequencies that we see, is with higher pitch, is a higher frequency. Lower pitch, lower frequency. And so what happens is those vibrations play a role as far as what we hear. We also notice we have to look at the volume, the intensity. How much do we get? So if we look at normal conversations at decibels, so we're looking at 60 decibels in a normal conversation, but 140 decibels would cause pain. 90 decibels is what the um, occupational safety um, o- organization will predict as being requiring uh, protective gear. So if you're looking at individuals that work in a environment where it has a lot of large sounds, that's when they would be required to actually have some sort of protective gear like earplugs or something else that would protect their ears. Um, in today's world, I have to say this that unfortunately, when we actually use earbuds and other types of devices like that, it actually generates the sound directly into the ear, and it may actually have higher sound volumes than are really safe. And so you'll notice, unfortunately, if we use these larger sounds on a frequent basis for an extended period of time, a lot of times we start to lose different frequencies in our hearing. And I know somebody right now, he has loss of hearing by 60% in one ear because of the fact that he was actually working in a factory environment for most of his life. And he has actually reduced the perception of certain sounds in that ear by that amount. Um, so when we actually also look at people that, like I said, I have students and I can hear their music from across the room if they're playing it with these earbuds. And unfortunately, that does have a long-term effect on them. Hopefully, they change their pattern. But you'll notice that even if you're actually having large amounts of that sound, it can actually cause deafness. Now, we have two types of deafness. We have nerve deafness, which is basically where we have damage to the receptors, damage to the cochlea, damage to the bones, and you'll notice that this will actually can occur. Now, nerve deafness would be more permanent in a real sense. Now, sometimes what will happen is it can be because of loud sounds over a period of time throughout the person's life. Sometimes it's related to, let's say, if they're in a environment where they hear explosions frequently, that would actually cause them the possibility of going deaf much more. Now, you might remember a guy by the name of Thomas Edison. You might also recall from history he lost hearing in one ear. Why? Because he was in front of the train on the tracks and he got caught there and he lost his hearing because the sound of the uh, the train um, horn was blowing directly into his ear, and it caused him loss of hearing in that one ear. So things like that happen. and sometimes there's no correction to it. We also have deafness, which is related to basically blockages or inabilities. Now, how can we do that? Well, as it says, it can be related to a perforated eardrum, okay, which may repair. It could also be related to infections like otis media, which is a middle ear infection, which a lot of times people that actually have ear infections, that's what we actually see, and it causes fluid buildup behind the eardrum, and it reduces the ability to hear or have the vibrations. Or it could be osteosclerosis, which is basically related to bone um, development in the ossicle bones, but basically you're not actually getting the vibrations. Okay, so conductive deafness can also be related to a buildup of wax. Now, here's one of the things: when you clean your ears, did you know you're not supposed to put anything larger than your elbow in your ear? Now, most of the time, people clean their ear with using Q-tip devices. And one of the biggest problems with it is when you're cleaning the ear, a lot of people don't realize they can push on the eardrum and they can perforate it. And it can cause damage to it. And you can also get the, uh, the cotton may fall off and cause a blockage. Here's another problem. And I talk about this, and I may have mentioned to you guys, but when we're looking at a lot of times during warmer weathers, a lot of times we actually get things that go into the ear that we don't expect. For instance, in emergency rooms every year, you'll notice in urban areas, there's insects that can crawl into the ear, like roaches. In more suburban and more rural areas, you may have more of the occurrence of bees or flying type insects that fly in. They crawl in, they see the space, they think it's a place to crawl into, and they don't know enough how to get out, and they get stuck. So that can also cause a conductive deafness. So basically, a lot of times the emergency room has to clean it out. Um, so it's not pleasant, but it is something that happens. Um, how do we get the physiology of the sound? Well, the membrane vibrates. It vibrates the round oval window, which then causes the membranes inside the cochlea to actually trigger the um, the cilia to move, and that cilia then triggers a specific nerve cell to then cause a change. That's basically what's happening. Okay, and it's all related to sodium channels, which may also have calcium involved to trigger that impulse. Like I said, pitch and volume, though, pitch is related to the number of vibrations. Volume is how intense that the sound is. So you might notice also when you actually go and you hear, let's say you go to a concert and it has a large amount of sound. You'll also notice that a lot of times you have ringing in your ear. And that ringing in the ear is related to having the vibrations that don't want to stop within the cochlea. And that can also, over time, cause some sort of nerve deafness. So, and basically, where does it go? The fibers will actually go into the temporal lobe of the brain until it gets to the thalamus and then to the primary auditory cortex in the temporal lobe. So, So when we also look at it, if we actually have nerve deafness, sometimes we can also have some patients that may be candidates for a cochlear implant. Now, what does a cochlear implant do? It's basically a microphone which is converting into some sort of electrical device, which, if you ever saw one, it sort of looks like a um, hearing device, but it actually gets translated or communicating into the brain. So it actually is directly connected to the pathways of the brain. Okay, but it's only a crude representation of the sound, so it's not as clear as what you actually hear without one of these devices. So I want you to realize that it's not exactly the same as having it. So one of the other things that we can see is osteosclerosis, which is basically an overgrowth of the bone, which is basically an arthritic type device problem. So we can actually have that and it reduces the hearing and causes hearing loss. Now, let's look at the vestibule again. Notice where we actually see the cochlea. Notice where we have the semicircular canals. Now it's the vestibule which will actually allow us to actually have the connection between these parts. And when we look at balance, balance is where we see the semicircular canals. So we have what I'm going to call rotational balance and and what I refer to as linear balance. So when we're looking at equilibrium, or in other words, balance, we have static equilibrium, we have dynamic equilibrium. Static equilibrium is what I usually refer to as linear balance, which means what are you looking at when your body is just standing there and able to get balance? Dynamic equilibrium is what I usually refer to as more of a rotational, which means you're moving, which is what you'll notice on the description. So, what you're actually seeing, if you actually, unfortunately, have those nice, lovely gentlemen that like to have these cars with bright lights on their roof, and they stop you in the middle of the night, and they ask you to basically stand there. Standing there will actually tell them whether you have static equilibrium. Walking that or walking that white line, that's more of the static equilibrium. If you're able to walk straight, as opposed to dynamic, which will be more of that, closing your eyes and touching your fingertip to your nose type thing. So that's where we actually see those two types of equilibriums identified. So when we're actually looking at it, you have structures that are involved in this, and we're not going to worry about these structures right now. But the real key that I want you to understand is that we have hair cells that are stereocilia or microvilli and kinocilium. That will actually allow us to have movements that has the fluids and the gelatinous membrane to actually get a triggering so that you can actually identify whether you're balanced or not. So when we're actually looking at it, your position of your head will actually identify the movements, which will go and trigger whether you're balanced or not. So just like this gentleman, he's actually moving his head. So when we're looking at it, they have three different directions, and the directions will actually, in the semicircular canals, will actually trigger the dynamic equilibrium, which will look more like this. So we actually also get hair bundles, which will also get triggered. And when we see this, you move your head, you move your body, and you get detection of it. And it triggers it to go into the cerebellum and through the motor cortex where we actually maintain our balance. Okay, so let's go on to review some of these things. With our eyes, we're actually able to actually get development of the eye. Now, the eye actually has two different developments, and I just want you to realize that it actually is an outburst of the brain development. So, if you looked at this diagram, it actually shows you how the eyes develop. And it's from basically two layers, the ectoderm and the basically the two layers of the ectoderm, from the brain tissue and the outer structure. When we get the ears, it actually is also a thickening of the ectoderm that actually allows us to create the the ear. which looks like this. Don't worry about the development at all. I'm showing it to you so you have a better clarification for your own understanding at this time. When we get older, we also have problems. Presby, sorry about that. The dog noticed something. So when we're looking at it, we get presbyopia as we age. We can get cataracts. Cataracts is where we actually have clouding up of the lens of the eye. And a lot of times that actually requires surgery. Now, years ago, they used to do conventional surgery, and the person would be actually hospitalized. And when they were hospitalized, it would take days for it to heal. And but today they actually use laser surgery for this. And so typically patients can actually have a normal doctor's visit, and the doctor's visit will allow them to actually replace the lens with a prosthetic lens. And the laser surgery allows a cauterizing of whatever damage was, so that actually reduces the repair and also the recovery period. So it's much better. Now, I do also know I came across somebody that actually required cataracts. They were not that old. And in the first, they had one eye that was had a cataract operation. And from their interpretation, having the cloudy eye with the cataract gave them more clarification and clarity into what they saw when they then after they had surgery. So it does actually happen. Your muscles of your eye get weaker as time goes by. So you actually have reduced ability to visualize. So that's why you actually may not visualize quite as well. Your tears will also decrease in their production, which also may dry out the eye. You have more of a possibility of retinal detachment or retinopathy and macular damage, which means reduction in your receptors, and more of a possibility of draining off the fluids, which would then cause more of a possibility of glaucoma. So over time, you lose your sight, basically. When you age, you also lose your ability to smell and to taste. Now, keep in mind, medications and what you do also affects it. So if you're used to eating a lot of highly spicy foods, that may also affect it too. But as you age, you also lose your hearing. So you may get tinnitus. Tinnitus is basically ringing in the ears, which is what you may notice if you actually go to a loud sound type experience. Uh, so I would say that part of the test, when you actually have the test, know about cataracts, know about glaucoma, know about color blindness, know about deafness, nerve deafness, and conductive deafness. Um, Meniere's syndrome, I wouldn't worry about. But otis media is one of those things that I would actually recognize. And most otis media, when you get an infection of the ear, you'll notice pain and discomfort, but it's in the middle ear. And usually, if you actually have a cold and you actually develop the ability to actually have that infection frequently in the same location. So, when we're actually looking at it, some of these diseases are really important to understand. Um, so, I will tell you with the test, make sure you know the two diagrams, the eye diagram and the ear diagram. Know about some of the diseases of the eye and the ear. Notice that when we looked at the smell and taste, they are integrated together and they actually affect your ability to distinguish flavors. So I want you to understand those. And so I hope that was helpful and I hope that was useful. So have a good day. I'll talk to you soon.